So I was first I came to the U.S. on athletic scholarship. I was a distance runner, and I got a 4-year athletic scholarship to run for this college in New Jersey. And I majored in computer science. So computer science was sort of like an afterthought. I actually wanted to major in exercise physiology, but they didn't have that major, so I was like, okay, I'll do computer science. And then I graduated in 1997, and I didn't want to go back to my home country, so I was looking for a job, and it was pretty easy to find a job as a software developer. I got a job as a software developer, programmer, software engineer, and I worked in this company for 4 years. But then I started getting a little bored with and I didn't see like really a future of like advancement was relatively limited. You can only become a middle manager type of position. So I was looking for a PhD to go into, and I found out that Mount Sinai you can get a PhD, and they pay for you to like have a salary stipend, which is completely news for me back then. I didn't even know Mount Sinai existed. I didn't have any biology background, but I had a lot of programming background, so I did a lot of coding and computer science. I also did a master's degree in computer science after I finished my bachelor's. So I was like, okay, I can combine computer science with the biology. And the first year as a PhD student, you're looking for, and also, a lot of you are master's students, you're looking for a project to do. So I was like, okay, how I can combine my computer science background with biology. So I was, interviewing different labs, and I went to Ravi Iyengar's lab, mostly a wet lab guy, but he was interested in computational stuff. And the members of his lab, they tried to create this signaling network. So even if now you go to the 12th floor of Icahn, that whiteboard is still there. You can see that maybe some people like leaned on it and stuff. It's slightly erased, but this is like a historical whiteboard that the members of the lab already, like in 1999, which is now almost 20 years ago, they tried to sketch the pathways, the cell signaling pathways from the membrane to the nucleus, like how cell signaling happens, where you stimulate cells and then the flow of information sort of go from the cell surface to the nucleus. So I immediately thought, okay, this is a network, and I can potentially analyze that network using these computer science methods that I knew about. And even though it's not really a network in a way that you have one node and one link, you have many copies of many of those receptors and things that are happening in a very complex way, and it's very dynamic, and there is a lot of components that are missing. So I took on this project and I started reading papers. So one of the things that I said to myself also, if I read a whole lot of papers and I'll figure out the connectivity, the wiring diagram that is inside a cell, and have all this integrated knowledge about how a cell works. So, how all those pathways are connected and how cells respond to a stimuli. So just to give you off the bat, I still don't know how it works, and that strategy didn't really work that well for figuring out how a cell works. But it was at the time the largest cell signaling network that people put together from literature. So we organized and visualized it like this. So we had a bunch of extracellular ligands that are known to bind to the receptors, and then those are connected to a bunch of like blob of cell signaling components, kinases and other adapter proteins, and GTPases that all are enzymes and complexes that influence the activity of different cellular machineries, such as transcription, translation, and the way I organized this information, I was like reading the paper and then decided whether there is a direct activation or inhibition of one node to another. And also, this person wrote review, news and reviews article that described that paper. And what they showed is that you can model the dynamics inside a cell at various scales and various levels of complexity. So you have like very low level complexity, which is like those molecular dynamics where you're looking at two molecules and how they interact, like the forces and stuff. And this is happening at a single molecule or single gene. And then it can be very precise. Then you have those ODE type of models that sort of like form here, where you have those circuits that have concentrations and how enzymes sort of like transfer information. And then you have this dynamical system that you can encode in ODE. And then there is a more simplified version of this, which is the Boolean networks or discrete networks, where you are deciding whether like things are either on or off. And then you have those gates, like AND gates and OR gates, that sort of like influence how the parent nodes influence your target node. And then the paper that we had, what we've done is this directed graph that was a cell signaling net that I showed in the previous slide, that fell into an even more abstract level, and that is we coined the term pseudodynamics. So the idea is that you introduce flows into this network and you see how the signals sort of like go downstream in steps. But in biology, the complexity is so vast and our knowledge is so partial. So I believe that the most fruitful research needs to be done even at less dynamics. So it's all almost like qualitative type of biology, but is more looking at finding which genes are responsible for which processes, and you'll see